Hello again, biology peoples. Mr. Kinnear here with another video lesson for higher biology. We're on Unit 1, DNA in the Genome, and today we're looking at mutations, which is Key Area 1.6. And this key area really connects what we've learned so far about genetics, about DNA, gene expression, etc., to what we're learning in the future. So Key Area 1.6, mutations, about the change to an organism's DNA, also leads on then to evolution and genomics, which is the study of the genetic information within an organism.
So to give you a definition of mutation at higher level, a mutation is a change in the structure or amount of an organism's genome, and that can result in no protein or an altered protein being expressed. So that's your definition at higher level of mutation. And the effect of that mutation will depend on its type and its location. And it can vary from a tiny change to the structure of the DNA to a large-scale alteration in the complete chromosome or the numbers of chromosomes. So obviously that's why we can have a wide-ranging impact of these genetic diseases. In our previous higher lessons, we've obviously talked about a protein requiring the correct sequence of amino acids to function properly. And if that base sequence is disrupted, then the amino acid sequence is disrupted, and that will cause problems for our secondary, tertiary, and quaternary structures, and that's therefore going to be problematic for the operation of that protein. But back in Nat 5, we also talked about things that could cause those mutations. So chemicals, mustard gas, nicotine, so mustard gas used in World War I, nicotine obviously present in cigarette smoke, but radiation, so gamma rays, X-rays, UV light, we're all familiar with those things. They are potential mutagens, so those things can cause mutations to happen. We'll now start to look at the types of mutations, and we've got two broad types. Single gene mutations where just a few bases are affected, or chromosome structure mutations where the structure of one or more chromosomes are affected. So a single gene mutation, fairly straightforward, where it involves a change in the DNA nucleotide sequence of a single gene. There's Amoeba Sisters GIF there, which are absolutely fantastic to help you kind of memorize and understand these three different types. So we've got substitution, insertion, and deletion, and I'll go through these individually to show you the impact they can have. So starting off with substitution, as you would expect from football or hockey, whatever, the substitute, you are replacing a single nucleotide in a DNA strand by a different nucleotide. So here we've got a sequence, our DNA, GTC, CTC, CTC. And what we've done is we've replaced that T with an A in our second codon. We've therefore got a different amino acid coded for, because again our mRNA code has been changed. A GAG in that second codon has changed to GUG, and therefore when it goes to the ribosome, a different tRNA molecule is going to be called for to bring a different amino acid into that polypeptide chain. So these are the least impactful. It's a minor change. It's often very small effect or no effect at all. However, you can get serious disease like sickle cell anemia if that occurs at a crucial point. We've got further classification now based on the effect they have on the sequence of amino acids in the protein synthesized. So I'll talk you through those. So three effects of substitution mutations here. Our first one is referred to as silent. It says it has no effect on the protein sequence. And as you can see, we've got a DNA string and then 4 amino acids. So obviously a very shortened sequence here, but it shows you exactly what we need to see. In our second codon, we've got GTA, which is coded for valine. And after a substitution mutation, we end up with GTT, but it also codes for valine. So remember from our translation lessons, there are more codes than there are amino acids. So therefore GTA and GTT both code for valine. So the protein sequence that we end up with is no different than what we should have ended up with. So that therefore is silent. It's not something you would notice within the organism because it's exactly the same. Missense is our second one. It results in an amino acid substitution. So here we have our third codon, CCC, coding for proline. A substitution mutation happens where we get ACC, so A has been substituted in for C there, and we get threonine in our protein sequence. So that's a substitution of the amino acid now, and again, that can have several types of impact. Some of our amino acids are quite similar, so therefore the impact can be very lessened. If you get an entirely different amino acid in there that doesn't have the right chemical structure for the right bonding at that particular place, then you're not going to have the protein folding at the right place and therefore impacting on the shape and therefore the function of the protein. So it can range from minor impact to major impact. And our last one is nonsense. That's where we substitute a stop codon for an amino acid. So that could be really problematic. So as you can see here, our fourth codon TAC is changed to TAG. So a G is substituted in for a C there. And instead of tyrosine coming in, we've got a stop codon, sorry. So that's where the ribosome will stop adding on amino acids. Now, if that happens very early on within our protein sequence, that's going to be incredibly problematic. Obviously, if it's towards the end, it might have a very small impact, but these can likely end up in a major change to our proteins. The next impact of substitution mutations is on splice sites. So that is where we have the boundaries between exons and introns. That means that it might prevent splicing at that site. So it could end up we're getting a massively different protein. Some introns may be included and some exons removed. So here's a diagram of that happening. So DNA sequence has exon, intron, exon, intron, exon, exon, and it should end up those two introns are removed. So we've got a sequence of 4 exons. We've got the correct protein synthesized. If because of our substitution mutation happening at that boundary of exon and intron, we end up with one of those introns not being cut out. So we end up with an extra bit of mRNA code still left in there. And obviously that could be a huge length of mRNA and therefore a huge length of amino acid sequence in there, which obviously impact on how the protein then folds and does its job. So this could be quite useful to get all that information down. So in terms of the classification of our single gene mutations, they're often referred to as point mutations because they happen at a certain point, obviously. So silent, it has no effect on the protein sequence at all. So obviously we're getting the same amino acid coded for, so happy days. Missense, change to the amino acid in the protein sequence. Nonsense, where we get a stop codon. So we've obviously got a protein that is shortened and a splice site. I just talked about it. You may get introns expressed and exons removed, resulting in a different protein. Our second form of single gene mutation is insertion. And as it sounds, we're going to add in one or more nucleotides into a section of DNA. So here's our original DNA, AGA, GTC, TTC, and you've got your mRNA code there. We end up adding in a G into our second codon. So instead of GTC, we get GGT. And as you can see, everything else from that point is shifted on. So remember, we've got 3 bases as a codon that will determine an amino acid. But now we've moved everything up. So actually from that point onwards, all the amino acids will be different. And that could lead to diseases such as Huntington's disease. Deletion is our third, and again, fairly straightforward in that we have removed one or more nucleotides from the DNA. So again, AGA, GTC, TTC, we end up with AGA, GCT, TCG. And again, we're impacting not just one codon, but all the codons after. So again, different amino acids from this point onwards. So that could lead to something like cystic fibrosis in particular cases. So these insertion and deletion mutations are referred to as frame shift mutations. That's because when the codons are read at the ribosome, the reading frame of those codons is shifted. So remember, it's reading 3 bases at a time. You add one in or take one out, then where all the rest of the bases sit is changed, either forward or back. And that can result in a completely different amino acids coded for. Or, as we've seen in our nonsense, it could end up with a stop codon being misplaced. So again, if that happens early doors, then it could be really problematic. So this can often lead to a non-functioning protein for those reasons. So a huge swathe of amino acids changed and or a stop codon misplaced, non-functioning protein results. So here's an example. We've got before and after, and as you can see, we should have started at CAT, but instead we've shifted down and we're now starting at ATT. And this C is obviously ended up as the 3rd base of a previous codon. So massive changes to that amino acid sequence. And again, massive changes to the amino acid sequence will obviously mean massive changes to the structural capabilities and the folding, etc., and therefore the functionality of that protein. So frame shift mutations, where the reading frame of the codons read at the ribosomes will have massive impacts on the proteins produced. So 3 examples of gene mutations and the diseases they can cause. You don't have to be able to quote this stuff, but it's worth having a look at these examples, BBC Bite-sized Scholar, to make sure you just understand they may come up in the context of questions, not that you'll need to remember, what causes these diseases exactly, but they might be referred to in exams. So cystic fibrosis, it's an insertion or deletion of the CFTP gene, so that protein can't be made. Muscular dystrophy, so impact on the DMD gene. So dystrophin is altered or not produced at all. And retinoblastoma, so insertion, deletion of the RB1 gene, so that protein can't be made. As I say, you don't need to know these off by heart, but maybe having an explorer of those through your textbooks, through the other information, so that you can maybe understand them should they come up in exam questions. So the second form of mutations is chromosome mutations. So that's obviously where we get changes in the structures of the chromosomes. So that can occur when the chromosome breaks. The cell then attempts to repair the break, but doing so doesn't restore it to its original structure. So imagine you're in a shop, you go careering down an aisle and you knock things over. You do your best to put things back as they were before, but maybe you didn't know what they were like before, so you just shove everything back. And that's what the cells do. They try and repair the DNA when they see breakages, but they could put it back in not necessarily the right order, and therefore that's when you end up with some chromosome problems. Just like single gene mutations, there are different types of chromosome mutations that can happen, but this time they can impact on many genes. And the 4 different types we've got are, as you can see from our great gift, duplication, deletion, inversion, and translocation. Our first form of chromosome mutation is duplication. So we're now looking at genes. So they're labeled here A to H. Sometimes they will be labeled numerically 1 to 10, whatever. So you should be able to identify whether it's a chromosome mutation or a single gene mutation. Single gene mutations will obviously have their base sequence, G, T, A, C, etc. But in duplication, what's happened? We've got DEF, it's broken away from the matching chromosome, so it's pair. And the cell tries to repair it, but what it's doing is it's putting it into the wrong chromosome. So we end up with DEF duplicated in there in the middle of that chromosome. Now that can cause problems. It's common in forms of cancer, but it also can be an advantage because we get 2 copies of that gene, And again, that will lead on to potential opportunities for evolution, but we'll talk about them later on. Deletion, as it sounds, we've got original chromosome that includes DEF. We get breaks around them, they become detached. Our cell does its best to solve the problem. It joins it back together, but unfortunately it doesn't add back DEF. So we end up with a shorter chromosome that's lacking those DEF genes, and obviously not able to express the proteins that are coded for by those 3 genes. The inversion, again, as it sounds, we've got a break and DEF is at the heart of it. Again, the cell tries to repair that, but as it does, it's rotated 180 degrees, and we've got the final chromosome that has got ABC, F, ED, GH. So that's a reversal of those 3 genes. Now that can be a problem when it comes to matching up with its partner during the formation of gametes. And it may end up with non-viable gametes, so you can't reproduce because this won't find its partner, because its partner would be the normal A, B, C, D, E, F, G, H, and this doesn't quite match up to that. Now, translocation involves 2 different chromosomes not matching pairs. So chromosome 1, chromosome 2, they're not matching pairs. So A to H and S to Y are different genes. And as you can see, we get a break in chromosome 2, and the cell does its best to repair it, but unfortunately what it's doing is adding S and T from chromosome 2 onto the end of chromosome 1. So we end up with translocated genes. And again, that causes us problems. So it leads to problems in gamete formation, so resulting in non-viable gametes. So a real impact on the potential success of this organism. So here's a simple table for our 4 chromosome mutations: duplication, deletion, inversion, translocation. That'd be useful to add to your notes. We've covered all 4 of those today, and obviously our 3 single gene mutations too.
And that finishes off our video lesson for today. We've looked at the fact that mutations are a change to an organism's genetic code. The impact of this change depends on the type and location of the mutation. Single gene mutations, which affect just one gene and sometimes just 1 or 2 nucleotides within that gene sequence. They can be substitution, insertion, or deletion. Chromosome mutations, so affecting more than 1 gene and a large part of 1 or 2 chromosomes, they can be duplication, deletion, inversion, or translocation. So hopefully you found that helpful. As always, if you have any problems, questions, or queries, get in touch, and I'll speak to you again soon. Bye.